So it is great to be here. I'm excited to talk about the work that we do at Morton Arboretum, the work that I'm interested in general. But it's certainly great to talk about it in a forestry group because ultimately I am most interested in forests and forest ecosystem processes. So I tend to start from the below ground, but my interests really sit much more at a whole ecosystem perspective. To give you a sense of my perspective, I did want to show you some of the background of where I really started thinking. So after I finished undergrad, before I went to graduate school, I went to work here. So this is in Central North Carolina, the Duke Free Air Carbon Enrichment Study, the Duke FACE site. I was tasked with monitoring below ground dynamics, root production, root turnover in this site for a couple of years. And I learned several things. I didn't know much about roots before I went there. Certainly, I learned all the good things that roots do. So they're important for resource acquisition, storage, anchorage. They play a large role in mediating the soil and microbial community and lots of other soil processes. But what really stuck with me and what has informed my perspective ever since then is when we looked at this site, which was many tens of millions of dollars to fund and run for about a decade, and then compared that to its sister site. So the Duke site was in Central North Carolina, dominated by pine. And this is the Oak Ridge FACE site, dominated by sweet gum. So two very different species, both important species. These had really different responses to the elevated atmospheric CO2 and the nitrogen deposition that was also crossed with that. And ultimately, they could not understand what was happening between these two sites without the below ground perspective. So really it was the different root responses and the different associations in their soil environment that I would argue dictated the responses of these systems to atmospheric CO2. So that has stuck with me since then. And even though I care a lot about the whole forest ecosystems, I come at it from a below ground perspective because I think a lot of our understanding the answers to our questions and how these species grow and compete and ultimately will respond in the future starts from that below ground, or at least our biggest gaps are in that below ground. So the research goals of my work are to understand these processes, starting again from roots, but their links to soils, microbes, and ultimately whole tree and whole ecosystem work. And as much as possible, I also do go out of my way to put my work in a context that is useful for modelers. So modelers at an ecosystem scale on up to a global scale. And I do this because I think it's important. I think it puts my work in a context that others can find useful, but also by working with these modelers, it helps me find out where the biggest gaps in our understanding are. So that's why I really put this as an equal emphasis in a lot of my work, even though I would not call myself a modeler. So what I wanted to talk about today are these two different stories. One is thinking about global root trait diversity, and specifically with some emphasis on gymnosperms. And then second is to talk about whole plant phenology. As a brief aside, I am interested in a lot of other things. In particular, recently, I've been interested a lot in trees and water, both trees and drought responses, but also trees and waterlogging responses, particularly in urban ecosystems. So if you're interested in drought or waterlogging, I'd be happy to talk with you all about it later on or tomorrow or send me an email. But for today's talk, I want to talk about these two things. And I do think that they really are related for understanding below ground strategies, for understanding how a whole tree does what it does. So when I think about traits, I'm really thinking about how can we define and easily quantify the function of these roots and the function that they play for a whole tree. When I think about phenology and whole plant phenology, at the root side, we're talking about when are those roots there to be functional? So these two pieces I do think go hand in hand. If we really want to understand below ground strategies of trees and ultimately how they relate to whole plant growth, we have to know both the traits that these plants are expressing, but also when are those roots going to be put out into the field. So on the gymnosperm side, a quick sort of plug for roots and root trait diversity. I think all of us can appreciate that there's a lot of diversity in the trees that we study. We see this every day when we look at the leaves. If any of us study wood, we also recognize lots of diversity in wood traits. But root traits have largely been ignored in a lot of this push to understand plant trait diversity. And I shouldn't say ignored because it's not really been a conscious choice so much as they're just really hard to study. So typically, if you do anything with a root, you've just killed it. And so you've prevented any long-term observations of that system. But when you do take the time, we have lots of diversity, both in form and function across different tree species, and even within species in that branched root system, or as that species changes, from seedling to saplings. So there's a lot to learn here. And certainly a lot of that diversity relates to the ultimate function, just like we know the diversity in leaf traits relates to their function. When I talk about traits, I'm a big tent type person. I don't know if you all are into traits and are into the debates about how people want to argue what the word trait means. I'm not really into that debate. So for me, it's anything that I can measure that tells me something meaningful for organismal performance. And really, just to go a step further, something simple, easy, and cheap to measure that tells me something useful that is often hard or expensive to measure is really what I'm interested in. So an example of that from the leaf side, some of Peter Reich's work, taking something like leaf nitrogen or specific leaf hair, which are very easy to measure, which can tell us something consistently about something that takes many months or many years to measure, like leaf lifespan. So my goal when I think about root traits is, can we do the same thing? Can we walk into a forest, dig up a root, make a very quick, simple measurement, and know something that otherwise would be hard or expensive to measure, like how long a root lives, or if that root has high metabolic activity or high uptake rates. That's really what we're trying to do. Now, root traits in general, this may not be surprising, but we have a lot of unknowns regarding basic trait variation, basic understanding of how different tree species do what they do below ground. But in general, if we can assess what they're doing and how those traits are coordinated, how they're related to one another, it should give us a good tractable way to understand the below ground strategies of these different species. We have lots of unknowns in terms of the basic patterns of root trait variation, just who has thicker roots, who has thinner roots. We're still, we still have a lot to open up in that box. And I would argue the reason is roots have largely been ignored from global trait syntheses. Again, not because people don't care, but because they've just been a lot harder to study. So when you go look, if you're into traits and you do plant trait analyses, you've almost certainly heard about the tri-plant database, tri-plant trait database. It was and still is the single best place to go for plant trait data. And they will freely admit they have terrible coverage of everything below ground. So in conversations with some of the folks who run tri, myself and my collaborator, Colleen Iverson, a few years ago, tried to put a lot of effort to actually pull root trait data together to say, hey, is there enough out there for us to start doing some of these global analyses to sort of pick up root traits and at least get them more on par with our understanding of leaf traits? So we did that by forming the Fine Root Ecology Database, which is horribly named or wonderfully named FRED, depending on how you feel about it. We first released this in 2017. We're now on our third iteration with FRED 3.0. If you're interested in root traits, please go here. So we continue to curate this database, and then we do actually directly upload this with the tri-plant database. So we have a lot of good crosstalk if you want to link roots and leaf traits together. So with the release of this database, there has not surprisingly been a pretty big boom in terms of global syntheses that have now come out in the past few years. So since 2017, I won't talk about coverage here, but obviously we have the same biases that pretty much every other global analysis has: great temperate coverage, weaker tropical coverage. But in the few years that we've had this, we've had a lot of really great studies, I think, that are pulling together these big databases, big data sets, to try to understand the global organization of root traits. Who has thicker roots, who has thinner roots? And I want to talk a little bit about some of the trait axes that we might care about. So the first questions that have really been asked are very, very simple questions. Things like, do we see the same relationships below ground that we see above ground? So typically we think of leaves as they're forming one single axis of variation. So pretty much the standard economic traits that we think about. So leaf nitrogen content, specific leaf area, leaf thickness, these things tend to fall out on one single axis. And we have basically termed this the leaf economic spectrum, where we have fast leaves, high metabolic rates, short lifespans, compared to slow leaves with slow metabolic rates, longer lifespans that really can be easily defined by things like leaf thickness and leaf nitrogen. So we wanted to ask the same thing. Do we find this single axis of trait variation that would relate to actual function of these roots in the root traits themselves? And the answer is no. So one take home is no, roots are more complicated than leaves, at least in this context. And we'll unpack that a little bit. So this is one of our studies led by Joanna Bergman. So just looking at the overall trait orientation, and don't want to get too into PCAs, though I will show several PCAs later. This is a cartoon of a PCA. But ultimately what we find is instead of all 4 of these traits, root tissue density, root diameter, root nitrogen content, and specific root length, these are the core economic traits that really relate to things like leaf nitrogen content, leaf tissue density, leaf thickness, specific leaf area. We do not see these fall off on one single axis. Instead, we see 2 distinct axes that are orthogonal or unrelated to one another. One of them was termed the collaboration gradient, basically based on root diameter and specific root length, that we think relates very closely to how much these plants rely on mycorrhizal fungi. So if you have big, thick roots, so high root diameter, you invariably have to invest heavily into mycorrhizal fungi because the roots themselves are not very efficient for nutrient uptake, but the mycorrhizal fungi are. So big, thick roots that are not efficient, you really have to invest in mycorrhizal fungi, as opposed to thinner roots with high specific root length. You can do whatever you want. You can have very little mycorrhizal investment. You can still have a lot if you want. You've got a lot more flexibility. In contrast, root nitrogen and root tissue density do not really relate to these 2. So what we're finding there is basically root nitrogen we still think is related to what we would call a fast root. So we think it is related to high uptake rates, potentially high respiration rates, though that's a little bit unsure, compared to roots with high root tissue density that we think have longer lifespan, so tend to be slower in what they do, but give you more return on investment over a longer time period. So 2 gradients instead of 1, 2 axes instead of 1 is what we see with roots. We did want to test and see how these related in a whole plant context. So this was a follow-up paper led by Alex Weigelt. And we hypothesized that we would see this conservation axis in roots mirroring what we see in leaves, and that these traits that actually at a whole plant level should all be related to each other. So root nitrogen should be related to leaf nitrogen. Root tissue density should be related to leaf tissue density or specific leaf area. In contrast, we thought that this collaboration gradient would not be related to any of the standard leaf traits. And then we did also look at plant height and root depth, which I won't talk much about now because we didn't think they'd be related to any of these traits, and they were not. So when we actually did this test, we found that our expectations were correct, that still the conservation axis of roots was indeed significantly, basically fell out in trait space right with those leaf traits. So root nitrogen, leaf nitrogen, root tissue density, leaf tissue density fell out very nicely. And in contrast, this collaboration accent still partitions completely separate from all the leaf and whole plant traits. So it is still consistent at a whole plant level. Roots definitely are doing something different. They definitely are a little bit more complicated, and it seems to be driven by the fact that roots have this association with lots and lots of mycorrhizal fungi in the soil, which seems to change their function, whereas leaves don't really have a partnership like this. There certainly are microbes on leaves, but nothing with the intense association like we see with mycorrhizal fungi. So that's what we see there. So with all that background, I do want to emphasize something is missing from all of these studies, either completely or partially. This is one of our studies led by Ma Ziqing in China. And I want to point out the families that he called out in this analysis here. So this is looking at the evolution of root traits over time. Basically, older families tend to have thicker roots. Younger families tend to have thinner roots. But in this figure, all the families that are called out are angiosperms, broad-leaved angiosperms. None of them are gymnosperms, because even though gymnosperms were in the broader analysis by Ma Ziqing, they were removed from this evolutionary analysis, and they were, generally speaking, weakly represented in the whole analysis. Similarly, this is work done by Oscar Valverdes. So this is the gymnosperm group right here, which, if you think about global diversity, gymnosperms are a species poor clade compared to angiosperms. So maybe this isn't unfair. But what I do want to emphasize, within the gymnosperms, we have high bias towards the ectomycorrhizal group, and very few arbuscular associated gymnosperms. However, when you actually look at gymnosperm diversity, so this is a family tree of all gymnosperms, with calling out different gymnosperm families here, all of them except for Pinaceae associated with arbuscular mycorrhizal fungi. So when you think about gymnosperms globally, they really are dominated by arbuscular mycorrhizal fungi. However, in our data sets, we have high bias towards just the Pinaceae. That's not bias for bad reasons. Pinaceae is undeniably economically, ecologically important throughout all of the Northern Hemisphere. But there are lots of other species. And then when you go to the Southern Hemisphere, we have lots of other groups that are also dominant and important. When you go to places like Southeast China, Kunihami lanceolata is really a dominant species and super important economically, ecologically, but wouldn't be captured by something biased towards the Pinaceae. So we wanted to address this. We wanted to address the fact that gymnosperms in general are largely absent from these global syntheses. And then what is in those syntheses tends to be highly biased towards this single family. No knock on Pinaceae, it's just there's more to the gymnosperms than that. Highlighting a few here, I do really like gymnosperms. I didn't really care that much about them before I noticed this gap, and then of course now I'm a big fan. But lots of weirdos like the cycads and neophytes. Certainly everyone loves ginkgo, and then I don't want to knock the conifers. They're still wonderful, but they are more than just the Pinaceae. We have the Cupressaceae, the Taxaceae. So the goals of this were really simple. Do we see the same trait organization in gymnosperms that we see in angiosperms? They have a very different evolutionary history. However, they are diversifying across the same planet with the same environmental constraints. So we could really expect this to go either way. So we started by quantifying the variation that we saw in gymnosperms and examining the correlations, because again, how those traits are arranged and how they're related to one another, we think gives us good information about how those plants actually have a strategy for acquiring nutrients in soil. So can we use this eventually to understand why pines are maybe good at some sites, but cedars are good at other sites? So we measured a bunch of traits. I'm only going to talk about the 4 that are highlighted here because these are the 4 that make up the core of all the other syntheses I just showed a moment ago. And the data that we used for this started with our fine root ecology database, but as we know, that's biased towards the Pinaceae. So we did specifically do several sampling campaigns, first where I work in the Morton Arboretum, and then with a collaborator of ours in Poland, Marcin Zaworny at the Cornick Arboretum. And then this spring, we took a sampling trip specifically down to the Montgomery Botanical Center in southern Florida. That was important because most of what we see in these northern temperate systems is still biased to all the same things that are in Fred. Still, we could go out of our way to get Taxaceae and to get Cupressaceae, but we're still missing 5 of the families, and there's only 12 extant families in the gymnosperm group. So we went down here to get the more subtropical and tropical dominating species. So Podocarpaceae, Zamiaceae, Cycadaceae, all those interesting species that I will say from a root perspective were a lot harder to sample. So we can talk about that later. So this is the data set that we came up with. I do want to point out a couple things. One, we now have 11 of the 12 extant gymnosperm families represented in our global database. The one we're missing is Welwitschia, which is exclusively found in the deserts of Namibia. So I don't want to knock it, but it's not globally, we don't feel terrible that we missed it here. The other families we have here are distributed globally. We do have a lot of Pinaceae, I can't get away from that, but this is representing about 1/3 of our data set instead of about 80% of our data set like we were in the other studies. So the first thing I want to point out is there's lots of diversity in the angiosperms. When you actually look at the traits and things like the coefficient of variation in the gymnosperms that we saw, there's no data here that's actually relevant, so I apologize for that. We see just as much variation in things like root diameter, root nitrogen content, specific root length, tissue density, just in the gymnosperm group. So it's a arguably smaller, more constrained clade, but we still see high variation at global scales like we do in the angiosperms. And then this very kindergarten-like PCA that I'll show here, just trying to show the alignment of these traits. We see basically the same alignment. So I want to call out the 2 main gradients on the right. This is from the Bergman et al. Paper, showing first the collaboration gradient, specific root length, really partitioning apart from root diameter. And we see the same thing here. And then we have root nitrogen content forming an axis again with root tissue density, which we see here. So even though there's a little rotation in these 2 PCAs, the alignment of all these traits is essentially identical between this global analysis dominated by angiosperms and this analysis dominated or made up entirely of gymnosperms. So this tells us a couple nice convenient things. The amount of variation we found in gymnosperms was quite similar to what we see in angiosperms, and how these traits are aligned is also quite similar. Again, we weren't surprised, but we were also a little bit surprised. So globally, the environmental constraints leading to the evolution of gymnosperms is quite similar. However, when gymnosperms have evolved into the diversity that we see today, happened at different times when the Earth wasn't the same. So we really could have seen the opposite response. We really could have seen different patterns, but we didn't. So this does give us some confidence that as we think about global scales, we can just assume pretty similar trends, pretty similar constraints on the gymnosperms that we see in a lot of the other angiosperms. So that's where we are there. This next part that I want to talk about is whole plant phenology. And again, I think the root traits, if you have high root nitrogen, that maybe means you have higher uptake of soil resources, higher respiration rates, maybe shorter lifespan. That's great. But ultimately, it still depends on when those roots are put into the ground that determines how efficient that root is, how effective that root is going to be. So I think these stories have to go hand in hand. So that's what I want to do next. And that's really where we're putting a lot of our effort at the Morton Arboretum for the next several years, because to look at things over time takes time. It seems a little silly to say that, but it's important to point out because you can't do a phenology study on roots in a summer. It doesn't fit in that time frame. So we have a lot of very basic questions. How variable is fine root phenology? What controls fine root phenology? And then I'll talk some about what we're doing at the Arboretum. We can't go into it today, so I just want to emphasize these two pieces and throw out some, I would say, thoughts or ideas or guesses about what might control fine root phenology, and then I'll show you what we're doing to try to address that. Now, when I talk about phenology, I'm talking very simply about the timing of resource allocation, allocation of those resources, and ultimately senescence. So when are leaves produced in spring? When does bud break happen? When is fall leaf senescence? And then basically the analogies of those questions below ground. When are roots produced? Ultimately, those roots have a lifespan, and then when do roots tend to senesce? Do they have an annual cycle like leaves do, or do they have a different cycle? Obviously, I'm biasing this towards deciduous trees when I talk about leaves and fall senescence, but we see, key phenological cycles in evergreens as well, so it's just as relevant. Below ground phenology is a big, wonderful open area. So I think there's a lot of questions to ask there because not a lot of work has been done. So it's primarily viewed from an above ground perspective. That's totally fair. Above ground phenology is still really, really important. And I'm not going to say that below ground phenology is more important than anything we have above ground. We know that when you think about the sequence of leaf emergence into spring, going to summer, and ultimate fall senescence, that defines your ecosystem productivity. That defines the uptake rates that you're going to have with photosynthesis in that system. Nothing about below ground will change that per se. But we do still have seasonal cycles of things below ground. We can have periods with essentially bare soil with nothing much happening, moving to periods of high root production. So I'm showing some lovely pine roots here with this nice red root. Ectomycorrhizas blowing outside of those roots and the fungal hyphae really attacking the soil. We see cycles of this, but we don't really have it quantified. We don't really know how species differ in this. So we have a lot of aspects of phenology that I think are important below ground, but we just don't know much about. Now, why might it be important? First of all, the job that roots have is to acquire resources that are not uniformly available across the season. So nutrients have seasonal cycles. Water availability has seasonal or sub-seasonal cycles. So what roots are actually doing is largely to tie into things that have seasonal cycles. So we would expect seasonal cycles or some type of temporal pattern with roots as well. The species that do this well potentially gives them a mode of adaptation to their environment, both as species move into different new environments, but also as environments become different and new for the species that are already there. And then ultimately, the ones that do this well gives them a way to compete with their neighbors. So if you are better able to tie into the changing patterns of resources in your system, you are going to have a competitive advantage over your neighbors. Now, those issues are all about what the roots do and what they take up. It is critical to also point out roots are incredibly expensive. So depending on the system that you're in, most temperate forests, we think about just the construction of roots taking somewhere between 10 and 30% of total productivity in a forest in any one given year. That's not counting anything about mycorrhizal fungi. That's not counting about maintenance, respiration, just the construction. And in many systems, this can be much higher. Whether you go into high alpine systems, you can actually have much higher amounts going below ground. When you go into grassland systems, upwards of 50, 60, 70% can go just the construction of roots. So this is a major cost. So actually getting the return on investment relative to the cost is going to depend a lot on how effective you are putting those roots out when the resources are available. So what controls fine root phenology? When we think about leaves, we know some things already. Things like day length, so photoperiod, temperature, both warming and the chilling requirements in many of our northern boreal systems. There may be other traits that have some relationship to the leaf phenology. So different types of wood anatomy may be prone to earlier versus later leaf emergence in the spring. On the root side, we don't have any of those quantitative fundamental understandings of what drives root phenology. But we do know some things. So we can talk about a few of these. I think it's fair to assume temperature and moisture are likely important. We know that they're major constraints, at least in temperate and boreal systems. And we do have some evidence for that. So this is a nice study by Birkenradel in the mid-90s. So we have temperature moving up here and perfectly tied in with that. Root production on the upper panel is also increasing. And you have peak root production and you have peak soil temperatures. In slight contrast to that, I want to show this study by Peter Reich et al. In 1980. So similar, as you're moving out of spring, you do see an increase in root production. But importantly, this is when we see root production in this site. But here, when soil temperatures are perfectly adequate for root growth, you essentially have no root growth later in that season. And the reason for that is because there's another factor here, soil moisture, that comes into play. So basically, as soil moisture tanks when you're in that hot summer period, you essentially have no root production. So it can't just be temperature. There's a lovely interaction here with soil moisture. Frustratingly, it gets more complicated. So pointing out here, lowest root production when you have lowest soil moisture. In contrast to that, this is another northern temperate site, southern boreal in southern Canada by Diego Steinecker. And we have the opposite pattern, highest root production when there was lowest soil moisture. So we can't just say as soil moisture goes down, root production goes down. There's certainly a lot of context for the site itself, but also the species that you're looking at that site. So temperature is certainly important, but it's not just a monotonic increase with temperature. Soil moisture is certainly important, but we can have complete opposite responses when you go from one site to another. Nutrient availability, I want to put up here. I don't have a previous study that makes any real convincing argument. I just think it's important since one of the main jobs of fine roots is to take up nutrients. And we know that nutrients, certainly of different forms of nitrogen, but also even phosphorus, which we tend to think about as being a little bit more stable or static. But phosphorus can also have clear seasonal cycles, pulses of high phosphorus availability. So it would stand to reason that this might actually play into how roots are responding to their soil environment. And the last piece I want to talk about is carbohydrate availability. This is the area that I think is going to keep us all employed for several decades trying to figure this question out, because it's much more complicated, but is ultimately, I think, going to be sort of the linchpin that helps us understand all these interacting factors. So I want to come back to this Reich study here, and you'll notice root production is going up here in April, but it really has this odd dip, even though soil temperatures continue to increase, and it's only in May that it really picks up again. So what I cut out of this figure is the timing of shoot growth. So there's clear competition within the system where root production goes up, but as soon as shoot growth starts, those two sinks are competing with each other, and shoot growth was clearly the dominant sink in this system. So it wasn't until shoot growth really finished that you had an increase in that fine root productivity as well. So we have to account for what else is happening in that plant, where else might that plant be allocating its resource, and among those different pools, who's the dominant sink at different times of the year. So at the end of this, where and when is each factor more important is what we have to figure out. And again, that's going to take a long time. So we're going to start off by just thinking about temporal trade-offs and how those manifest across leaves, stems, and roots. We're going to simplify this. Just think about three tissues. We're ignoring fruit production, seed set. We're ignoring flowers, just these first things. Don't know what that extra S is for. So here at the Morton Arboretum, we're going to think about and monitor explicitly the timing of leaf, stem, and root production together. So this is where I work. It's a public garden. It's about 1,700 acres. So it's quite sizable. Within that, we have natural areas of somewhat unmanaged forest with oak, sugar maple, cherry. And then we have lots of showy areas with beautiful trees from around the world. And then we also have a lot of forestry plots. These plots were established in the 1920s and 30s to figure out what trees would grow best in the Chicago region so that people could use those for timber production or different forestry purposes. By the late 30s, it became very clear Chicagoland is not destined for timber production, but destined for a lot of urbanization, suburbanization, if that's a word. So they stopped planting these, but they left what they had there. So I get to walk into this and have all these nice monospecific plots, which make it much easier for me to measure anything below ground. So as a root scientist, if I can go into a plot, dig up roots anywhere in that plot and know pretty confidently what tree that belongs to, that simplifies my world tremendously. So we get to work in plots like this. This is our Picea abies plot here on the west side. Nice monospecific plot, open area, understory. From that, I can easily monitor leaf and stem phenology, and now I can also easily monitor root phenology. So we do that using mini risotron cameras. So a mini risotron camera is a fancy name for a cheap, clear piece of plastic tube that we install into the ground. And then we put an absurdly expensive camera that is just a fancy desktop scanner into that tube. We come back to that, we scan the soil environment every 2 to 4 weeks and basically see when new roots come in. And from that, we can track when are roots being produced. And then we follow that across many months and ultimately many years to see when those roots die and how long they live. Importantly, we are doing this as much as we can throughout the entire year. So most previous mini risotron studies stopped in the winter because it's annoying. And there was a very prevailing assumption in the literature that roots generally weren't produced in the winter. And certainly we know that there are more roots produced usually when it's warm. But for several of our species, in particular our gymnosperm species, we have seen pretty significant root production right in the middle of winter, particularly at depth. So once you go below that 10 centimeters, the soil environment can still be adequately warm for production. So we have put a lot of effort into this, which is unsatisfying for Bill, but very satisfying for science. Stem phenology, we're using digital dendrometers. These give us a really wonderful daily to seasonal record of stem expansion. So we are focusing on expansion. We don't necessarily have data on biomass infilling within the stem, but this gives us our first snapshot of stem phenology. And then leaf phenology, getting continuous records with phenocams. So just taking images every day, and within those images, we can track the greenness of the canopy as new leaves are coming on, as old leaves senesce and fall off, gives us a nice continuous record. I mentioned models earlier, and that really has directly informed what species that we use. So my goal at the end of this is not to say, I can tell you all about root phenology at the Morton Arboretum. I want to specifically be able to say, this is root phenology that is typical and representative of temperate forests across the globe. So we've explicitly gotten a phylogenetically diverse, functionally diverse set of species. Within the angiosperms and gymnosperms, which we have both nicely represented, we again have roughly equal representation of arbuscular and ecto-associating. So we're not just getting only pines in our gymnosperms, trying to get some diversity in our wood anatomy. Other functional traits like growth rate, all those things. And I do want to emphasize here, we didn't do this at first. This was a bit of an oversight. So after a year, we added these in. These are deciduous gymnosperm species because we basically, our first data that I'll show you today is directly conflated when we think gymnosperm, angiosperm with deciduous and evergreen. So we added these. You won't see a lot of data from that today, but we're going to tease that apart later on. So couple things to point out here, sort of an awkward, colorful graph here. These are root phenology patterns for 18 of our plots in the Morton Arboretum from 2019. There is a lot of variety across them. There are some species that have their peak production much earlier in the season here, like July, much later in the season here, like October, November. A few species had a little blip coming up early in the season before leaf production. A lot of different stuff going on here. But let's pretend like we're modelers again. A modeler is never going to care about idiosyncratic species-specific responses. So can we tractably simplify this in a way that modelers could use? And the first easiest thing that we can do is think about it in angiosperms, angiosperm, gymnosperm. Not really sure what happened there, but in general, we see that even though gymnosperms are the ones prone to that early season blip in this single year, angiosperms on the whole seem to have earlier peak root production compared to our gymnosperms. Gymnosperms are the ones doing that late fall production, moving on into winter compared to the angiosperms. However, I would never tell a modeler to put this into a model from 1 year of observation. So we really have to do this, show that this is consistent across multiple species, show that this is consistent across multiple years before you put this into a global model that's going to predict future climate. And again, this was perfectly conflated with deciduous and evergreen. So keep that in mind as well. We'll be separating that out in the coming years. So looking at now how roots, leaves, and stems relate to each other. So this is one example here from Ohio Buckeye, Aesculus glabra. And so we're showing the timing of when production was most active for those species. This is spring leaf out when greenness was increasing. And after the leaves are finished being produced in that year, only at that point do we see stem expansion pick up. And only after stem expansion is done do we see our root production pick up. So we have clear sequence of events here, and we have clear asynchronicity across all of those different tissues. So they're not happening at the same time. I want to emphasize that because almost every ecosystem to global scale model assumes that root production and leaf production happen at exactly the same time. That is something that I would argue we can put into a model today, and I feel very comfortable with. This general sequence was pretty consistent. There's noise when you look across multiple species. Everyone has their own thing that they want to do. But in general, this sequence of leaves to stem to roots, leaves to stem to roots, leaves to stem to roots was consistent across essentially all of our species, including our gymnosperm species. So again, leaves to stem to roots here in Picea aves. So despite the fact that there's always something unique happening, that general sequence was quite consistent. Now, I do want to remind you all, in some of our plots, we do see early season root production that happens before leaves, and that's what Peter Reich saw in his study as well. And I've actually seen that in other studies before in some of my other work from other sites. So there is potential for early spring root production in most of our species, and we'll talk a little bit more about that in a moment. But we're not showing this in any of the examples here. Ultimately, our next step is to think about this. What are the drivers of the differences, potentially across species, but as we add in more years of observation, what are the environmental drivers driving that potential spring blip of root production for some species in some years, but not others and not in all years? So we do have all this stuff measured, so we can do this. We just need more years of observation to do it. So all the environmental correlates you might dream of, we have most of them. We can't really do much quantitative with that at this point, but I do want to show you an anecdotal story, just comparing the year 2019 to the year 2021. So I've split up my angiosperms and gymnosperms here, and this dotted line in all of them will be July 1st. So in general, we see that in 2019, essentially all of our species had their peak root production around or well after July 1st. Both angiosperms and gymnosperms. Again, maybe notice the peak here is a little bit later than sort of the weight of the peak in the angiosperms. When we compare that to 2020, there's a strong shift in essentially all of our species that was consistent in both the angiosperms and gymnosperms to have peak production around or before July 1st. The big difference between these 2 years was 2019 was incredibly wet. Soils really had that sort of spring snowmelt saturation that usually goes away sometime by late May. The snow melted, but then we kept getting rain and rain and rain. We had a lot of flooding, and our soils really had tremendously high soil moisture contents well into the middle of the summer. In contrast, spring 2021, snows melt, really no precipitation after that. Soils were already beginning to dry down to sort of typical late summer soil moisture levels by the time late spring, early summer was coming around. So very dry spring, very wet spring. And we might argue here that that wet spring really delayed and pushed a lot of production back in 2019. There's nothing quantitative about it, lots of hand waving, but I feel reasonably good about sort of the trend that we might see there that hopefully will confirm with more years of observation. So summary there, we see certainly a lot of variation in fine root phenology across our species. There's maybe some tractable distinctions across plant groups, but again, I really want to confirm that with more years. The trade-offs in resource allocation, this clear asynchronicity has popped up in tiny snapshots, like some of the earlier work from Peter Reich and lots of other forestry studies. And we see that with overwhelming confidence in our species here. Asynchronous production across these dominant tissues. So really, there has to be a trade-off competition among those species. So again, I think the carbohydrates, the non-structural carbohydrates, where and when they're allocated will be a key piece of this story. And then from year to year, we certainly are going to see, at least we have seen strong year-to-year variation. Quantifying that and predicting why certain years have earlier versus later phenology is the next piece that we want to do. So to do that, obviously, we have some fun quantitative analysis to do. I want to add in mycorrhizal fungi because I'm a root person first, but very closely after that is acknowledging that mycorrhizal fungi are really critical to the strategies that plants have below ground. So we're going to start adding more explicitly quantifying that mycorrhizal component in the next couple of years. And then just as a thing that I really wish I could do is more disturbance trials. So again, thinking about how the carbon balance, the non-structural carbohydrate pools of the whole plant change over time. We know things like spongy moth outbreaks, strong summer droughts, those have strong effects on carbon balances of the whole plant, which have the potential to really wreck the sequence that we see in sort of typical good years. So that's something we'd actually, through multiple years of observation, we're going to get a few lucky, sorry for the plants, but for science, lucky disturbances, but actually doing some controlled trials would be a lot of fun in the future. So that's all we talked about today. Traits, phenology, both of those together define, I think, a plant's below ground strategy. They go hand in hand. I'd love to talk about all of this. I really am passionate about the work we're doing at the Arboretum, so I'd be happy to talk to any of you at any point. We are looking for a postdoc for this fungal side. We do have a grant-funded postdoc to start actually thinking about how these fungi play into the overall whole plant phenology of these different species. So feel free to reach out to me very soon if you're interested. Otherwise, I'd be happy to take questions now. Thank you for your time, or reach out to me over email in the future. Thanks. Yes.
We were talking about ingredients and we're thinking about some of the invasive weed species that really change in the neck. And they're really thick, or we should say, high density fine root systems. It seems like they have when you yank them out of the ground. And if there's any relationship there between that and their allelopathy or their high competitive nature, so it makes them invasive.
Okay, so I'm going to attempt to repeat that for the online group and then give you a really unsatisfying answer. So in general, like invasives are a problem. Invasives are super frustrating, and a lot of them seem to have distinct, I'll even say unique below ground strategies in many cases. Think about garlic mustard, which we think mycorrhizal fungi are super important, and they don't use mycorrhizal fungi. So there do seem to be a lot of the invasives that come in and disrupt the system because they aren't necessarily following some of those trends. And so because of that, I use the word unique. My sense at this point is that I don't see invasives consistently falling on one side, like, the fast, aggressive, high turnover, but high competitive sense. I don't see them always manifesting that root trait strategy. Because some of them do have fatter roots, some of them may manifest it through this allelopathy that does not fit into these 4 core economic traits. But I think that's super important. Actually, I've I just spent an hour talking to you about, or at least half hour talking about these 4 core economic traits. And I really, really find that unsatisfying to a certain extent, because stopping there is what I'm afraid that other people will do. Root hairs, actually quantifying the function of mycorrhizal fungi, and then all these other chemical attributes like allelopathy that really are going to change how that species does what it does, or potentially how it manipulates what someone else does, are all really, really important. So I think these 4 traits are a starting point, but they don't capture things, ways that I think a lot of our invasives are actually succeeding.
Really a lot of interesting points there. So the delayed production of fine roots by the gymnosperms found in the evergreens, is that possibly linked to late season or winter photosynthesis? And do you have evidence for that?
So the question is, could the delayed production, this is really fun, bio. Could the delayed production of the gymnosperms be related to late season photosynthesis? Is that correct? I think so. So I don't have evidence other than we know that after deciduous trees drop and you get those beautiful fall November days that are just, there's nothing limiting photosynthesis at that point. Yeah. Yeah, a lot of our deciduous species are in senescence and aren't doing a lot, yet the gymnosperms are doing fantastic. So I do think so. I don't have any measurements that actually would confirm that. I don't have, tracking of late season sugars that are developed going to roots, but I can't imagine that is not one of the reasons why we get longer late season root production in some of those gymnosperms.
Unrelated question. The chronology of first the leaf production and wood production, then the root production. And in the work that I've done previously on non-structural carbohydrate storage, kind of the most it's been well characterized, but kind of the model at the time was that the plants store carbohydrates after building structure, so that it would happen after all those other.
Mm-hmm.
Do you think that it would happen simultaneously with root production or after root production? Does that seem like a reasonable operating perspective?
I think it's a reasonable. Oh, sorry. Trying to recapture that. So I got really interested in the question, so I kind of got to summarize it now. So thinking about non-structural carbohydrates, the data that I showed had leaves, then stems, then roots. And basically, where would the buildup of non-structural carbohydrates fit into that sequence? And is it potentially likely happening during that time when root production is highest? Because what we do see typically is that non-structural carbohydrates are building in late summer into fall before leaf off. So because we've seen those patterns of non-structural carbohydrates, I think the answer is yes. They must be happening to some extent simultaneously, because I see root production in August and September, and we know from other studies that for a lot of our deciduous temperate forests, carbohydrates are usually, critically are building up quite rapidly during that time. So there has to be, to some extent, they're happening synchronously. So if I plotted those on the same plot, we might expect to see them basically happening at the same time. But I do think there's additional questions to that is, for example, I tend to think at this point, I think increasingly roots will be produced at any time as long as nothing else is telling them they can't. So if the environment is favorable and there's not a competing sink that is going to outcompete the roots, I think they'll happen. And so if the weather was really bad in fall, I suspect we'd see a strong dampening of root production, but maybe not of non-structural carbohydrates, because if you're a birch, you can't let that go. But you can maybe let go some of the luxury production of roots, if that is indeed luxury production, which is a whole nother set of theories. So I do think they're synchronous, but there's still additional questions there of who's the priority during different times of the year.
Can you take a look at the
Time?
and
Oh.
get a couple of questions? You can read
I absolutely try.
Chat, in the bottom, in the middle. Yep, over there.
Okay, yes, you hear me? Have you obliquely, so in the first question, so in none of those PCAs was there any rotation at all. I'm not making a political statement for or against rotations in PCAs, but none of ours were rotated. Here in the West, pine seedlings are not infected with EM. Within a year, they die. The same in the East. I mean, I think yes. I think most pines pretty much have to have EM. We've gotten them to grow them in a greenhouse for 6 months or so without mycorrhizal colonization. But as far as I know, I don't think many pines survive in the wild without colonization. That's my shoot from the hip response to that based on literature. I've certainly not looked at it myself, but we see that in lots of cases and in lots of really cool invasion questions. Pines around the world seem to need the colonization. So certainly in the East, they're colonized, and my sense is they wouldn't succeed without that. But Aki or others might know more about that. Are there any other questions there? I think that's it.
So I got a question. So it's about the your experiment at the modern aboriginal, and you compare the above ground and wood production. And it's you can capture the dynamics of the above ground and below ground, but there is that instrument sensitive enough to capture the wood production? The instrument that's based on the side, is it good enough?
Yeah, so yes and no. They are great. I'm a huge fan of particularly the Thompson point dendrometers. If anyone wants to hear me wax poetic about an affordable but good tool, I would point you to that. So they are really, really sensitive down to microns. We get beautiful patterns of daily shrink swell that can help us diagnose other things like drought stress, in addition to the slow, steady incremental growth across the season. So in that sense, yes, they are definitely sensitive enough, but they do not tell you anything about biomass infilling after swell. So, we have files laid down in the spring, and then what we're capturing really is the swelling of those cells with stem expansion, and then capturing when they don't shrink back down with drought or something. But we're not necessarily capturing the thickening of cell walls behind it. So there is a second part to a stem growth story that is all about that biomass infilling that we're not capturing. But I do think we capture really fantastically that sequence of expansion across the season, which is also important for the carbohydrates. So it's not, I don't think we're missing the story, but we are not getting all of the story. Yeah.
So the pattern, sorry if I can't articulate this well, the pattern of species allocation, like leaves to wood to roots, we sort of break it down between angiosperms and gymnosperms, and angiosperms are predominantly deciduous, gymnosperms predominantly evergreen. So what would you expect to see in like a evergreen angiosperm like ilex, or and talk about the deciduous gymnosperms with things like lumerics and things? Would you expect this to be more a difference between evergreen and deciduous, or a difference between gymnosperm and angiosperm? That kind of makes sense.
Yeah. So, and I forgot to repeat your question for the people on, but so I showed a lot of difference between angiosperms and gymnosperms that seem to be related to different timings of leaf, stem, and wood growth. And what I didn't show you all is the new data we have for our deciduous gymnosperms. But then you also brought up the issue of like, what about evergreen angiosperms, which we completely lack, which, trust me, I walked all over the Arboretum trying to find some good ones. So would that be important? And ultimately, what is going to be more important, that phylogenetic signal of angiosperms versus gymnosperms versus the functional manifestation of a deciduous species versus an evergreen? Certainly the answer is I don't know, because I don't want to give away the big story that hopefully I'll have when we finish these data in a few weeks. But in all fairness, I don't know. If I was guessing, I think that functional leaf habit is likely to be more important. So, and I will say, I looked all of the arboretum, and then we also put in a grant to get ilex and at another site, and to also get some southern species. So we do have Magnolia grandiflora and florifolia. Feel like, you know, biffing that in a forestry department is not the right thing to do, but so getting some of those evergreen angiosperms, to answer it, we'll have to measure those, but we just don't have any, you know, north of the Mason-Dixon line. Beyond that, I also, you know, I really emphasize temperate boreal systems. But once we go into a tropical system dominated by evergreen angiosperms that we know, you know, notoriously are difficult to age because growth rings don't happen in the same way that they do here. Like, I think all bets are off, and we'll have new sets of constraints. So here, temperature, that seasonal cycle is critical. There, maybe we're going to see those cycles driven exclusively by water availability. But in that case, leaf habit doesn't matter as long as the leaf habit is tying into the water availability. Just a guess.
Just last question.
Yes. Oh, this is much easier. Is the time series of growth versus taxa you have run a PCA? I'm not sure I understand that question. In fact, I'm not going to embarrass myself by trying. If you can clarify that, I will try, but I may just not understand.
More question? Okay, thank you so much.